Hi guys. I'm gonna make a quick video going over. Well, I don't know if it's gonna be quick, but I'm gonna make a video going over adaptive and innate immunity. Going over innate immunity first, and then adaptive. And just already off the bat, the slides that I'm gonna be using are something that I made on my own in like 2 seconds, so they're kind of bad. But if you guys want them for some reason, then just let me know in the chat and I'll post them. But before we go over in immunity, we have to go over the basic steps of the inflammatory response. So whenever you have a mast cell identify a pathogen, I'm going to go over what a mast cell is. But whenever you have a mast cell identifying a pathogen, which usually happens in an area where you have like a break in the skin. I don't know why the skin shook like that, but anytime you have a break in the skin like that, then you're going to have pathogens entering your body. So what happens first is that mast cells, where are mast cells? Here. Mast cells are gonna degranulate, and that essentially means that they're gonna make vasodilation occur, which is like this right here. They're gonna make the cells in your bloodstream separate so that they can give way for the other immune cells to get to the area that's usually infected, because the area that's infected is not an area in your blood. It's closer to the periphery. So mast cells are usually in the periphery. Then after that, once those cells start getting into the area, they let out chemokines, which make other cells go into the area through chemotaxis over here. That's the second step. The third step is that selectins make neutrophils move along the endothelium. So that's this picture right here. The selectins kind of like drag the neutrophils along the endothelium until they get into contact with a chemokine, which makes them move into the affected area. Then neutrophils penetrate the tissue and then they start eating the things. So this would be acute. The main difference between acute and chronic is that acute, once you do this, you kill off the pathogen. With chronic, your cells aren't able to kill off the pathogen, so what they do is they just wall it off. And the way that I remember this is that bees, when they have a bug that's like, let's say this is the size of the entrance of the hive, they have a bug that's like this big. That bug can't fit through over there. That would be like our pathogen that our cells can't kill. Because that bug can't fit through that little hole, what the bees do is that they surround the bug, they like move a lot so it cooks the bug to death, which is kind of graphic, but hear me out. And then what they do is they put beeswax over the bug, which kind of walls it off. So the bug never leaves the hive. It's just, it can't do anything because you have a bunch of like, it's dead. So in chronic inflammation, technically the pathogen cannot die. It can just stay dormant for a little bit. But it's the same idea. You're walling off the pathogen so that it can't get into any of the unaffected cells. But you don't necessarily kill it. You wall it off. So you have certain chemical mediators that are, oh yeah, and the name of this, my bad, the name of this would be a granuloma. So just so you know. So if you have a granuloma, you have evidence of a chronic infection, and granulomas are permanent tissue damage. That tissue is never going to be useful anymore. Okay, now we have certain chemical mediators that you use to protect yourself. So you don't only use the stuff that your cells make. Well, these are made by your cells, but like you don't only use the actions of your cells, you also have like some little chemicals that help. One of them are lysozymes, and basically what they do is they go where the nag, I can spell, where the nag meets the nam, and they break it off. And as you guys know, if you break the peptidoglycan of a bacteria, the bacteria is going to die. So if you think about it, which bacteria would this be more, would this have a higher affinity towards? if you don't know the answer, just think about the structure of different bacteria. So if you have a gram-positive bacteria, you have a bacteria, and then you have a thick peptidoglycan layer. And if you have gram-negative bacteria, you have bacteria, a smaller peptidoglycan layer, and then an outer membrane. This outer membrane right here, the way that you get in through it is through porins. So this will make the lysozymes have like a little barrier. So they have a higher affinity for gram-positive bacteria. They attack them easier. You can also have lactoferrin, which I don't have a picture of here, but basically what it does is that it takes up iron so that the bacteria can't then use that iron for their own metabolism. And that's basically all they do. And then this right here, this is a defensin, which is a type of cationic peptides, but cationic peptides, they're secreted by immune cells. And basically they just like damage bacterial plasma membranes. There's one type of them that damages the mitochondria, but like if you just know that they damage bacteria, you know enough. You don't have to know the different types. But the most important part is this complement system. So the way that you activate it, you have 3 different ways. Ignore the explanations given in here because these are not according to what we learned. But basically the classical pathway uses antibodies. So it's activated when antibodies come in contact with a pathogen. There's the lectin pathway, which happens when lectin binds to mannose sugars. And mannose sugars are something that you see in fungi. So that's basically for attacking fungi. And then you also have the alternative pathway, which is just anything else. So like LPS or any surface antigens, that's the alternative pathway. So basically, if it's not mannose and it's not antibodies, it's the alternative. And now the important thing is that we have 4 different complement proteins that we are going to focus on. First, you have C3A and C3B. C3A, if you remember in the animation in our homework, it specifically helps with also with not necessarily vasodilation, but it helps with like separating the cells in the blood vessels to make the blood cells go to the actually affected area. It just makes the inflammation worse, which you'd think is bad for you, but no, because you're just bringing more cells to the area. That's good. C3b, it does something called opsonization, which is basically like if you have a bacteria and you coat it with C3b, then it's easier for like phagocytic cells to notice it because it's almost like this is sounding an alarm. However, C3b is not the only thing that works with opsonization. IgG, which is a type of antibody, can also coat bacteria with C3b. So if you have IgG alone, that's okay. C3b alone, that's okay. If you have both of them together, then the cells are gonna be like, oh fuck, I gotta eat this bitch up. So they like increase the other's effect by like a shit ton. They're really good together. You also have C3 can also turn into C5, and C5a also induces more inflammation. So just think of the A's as like the kind of useless ones. They don't really do anything directly to the pathogens. They just kind of call the other cells. And then C3b forms the membrane attack complex, and it forms that with C5b, my bad. It forms that with C6 to C9. I'm so sorry, C9. And then it basically forms little holes on the membranes. Now, what else affects something similar to a membrane? What else affects something similar to a membrane? Not sure if you guys remember, but when I mentioned lysozymes, they don't affect the membrane, they affect the peptidoglycan layer. This membrane attack complex affects the membrane, almost like it's in the name. So which bacteria would it be easier for it to affect? Gram-positive or gram-negative? I'm sure you didn't answer that, but it's gram-negative because they have this outer membrane that's readily available. So they can come in, fuck this up, and then fuck this up. But gram-positive bacteria have this thick peptidoglycan wall, and they can't really get through that. So this works better for gram-negative bacteria. So that's it for the complement system. I will briefly come back to this when we get to a figure that was in the PowerPoint, just because I don't want you guys to be confused about that. So now we're going to talk about cytokines and cell progenitors. So you have 5 different types of cytokines. These are called colony stimulating factors in our PowerPoint, but you shouldn't really care about them. So the ones that I would focus on the most are interleukins and chemokines. Chemokines, basically, they cause chemotaxis. It's almost like it's in the fucking name. And when chemotaxis occur, the immune cells move to a specific damaged area. So they're very important for immunity. They're like step 2. And interleukins are just how leukocytes interact with one another. So when we talk about like T cells activating B cells and activating like cytotoxic T cells into differentiating, that happens through interleukins. So those are the main 2 that I would focus on. Tumor necrosis factors and interferons, in the PowerPoint, they kind of have the same function. I just know that like this one's antiviral, this one is like anti everything, I guess. And then colony stimulating factors, it stimulates the cell colonies to proliferate and differentiate. Almost like it's in the name. So I wouldn't really focus on those. They're easy. Now, let's talk about the different cell progenitors. These cells right here have a different progenitor than these cells. Nope. Than these cells right here. Pretend that NK cells are here. Sorry. These cells, they're kind of like simpler than these cells. That's how I remember it. Or you can also remember these are the innate immunity one. And these are the adaptive. I mean, except for the NK cells, but you know what I mean. The NK cells, I already grouped them with cytotoxic T cells because they're just very similar. They both do the kiss of death thing, just in slightly different ways. So that's how I remember that they're from the same progenitor. But basically, the innate cells come from myeloid cells, and the adaptive cells come from lymphoid cells. I'm not writing that down. You heard me the first time. I'm lazy. So now we're going to talk about all the different cell types. And I will say, if you're weird and you're a nerd and you don't mind being weird, there's this anime called Cells at Work, and it helps you remember what each of the cell does because they have all very distinct designs. So I'm not going to reference this. I'm only going to reference it once because it just, the association, like if you see like a picture, like of a diagram of blood cells, then the association is going to help you. But like, you don't have to, like, no one's going to test you on Cells at Work. I'm just, if you're a visual learner, then it might help you. So mast cells, they have 2 very special things about them. They, as I said, they degranulate, which controls vasodilation, but they are also coated with IgE. If you guys remember from class, IgE is an immunoglobulin, which is an antibody. So basically what happens is that they kind of work like B cells. They don't turn into plasma cells, but what they do have is that they're coated with IgE. And then, but instead of being coated with like a single type of IgE per cell, like the B cells are, each mast cell has like different types of IgE on them. And then if one IgE binds to an antigen, then the cell is like, oh, I have to degranulate because there's some weird in this area. That's why the IgE is there. So that's why they're useful. They sound the first alarms. Granulocytes, wouldn't really focus too much on them. These two are for allergic reactions. And then this one specifically, it's used for against like parasites. But again, they're not really involved in like a general inflammatory response, so I wouldn't focus on them too much. Neutrophils, however, this is when I am going to reference back to the cells that work thing, because if you ever see like a blood diagram, you have like a bunch of red blood cells and a bunch of white blood cells. If you think like average white blood cell just eat stuff and kill it, that's what neutrophils do. They just be eating shit. They can't present antigens, but they're really useful because they're like great at like killing pathogens. And that's like, you want the pathogens to die. That's pretty good. Macrophages, they're like neutrophils with a twist. So they are also highly phagocytic. However, they have MHC2. Holy shit, I can't write. They have MHC2, which means that they can present antigens as well. Just like dendritic cells. They also have MHC2. NK cells, they kill infected cells through like receptors that they have on their membranes. And they do it by they either like do the kiss of death thing where they like make like kind of like a cell suction cup between the NK cell and the cell that they want to kill. And then they just inject it with like, what are they called? Oh yeah, they inject it with perforins and granzymes. The perforins are the things that make holes in the membrane, but this isn't just to kill the cell. This is so the granzymes can get into the cell, and what the granzymes do is that they induce apoptosis, which is mediated cell death. So you're basically like, they basically like commit assisted suicide. Yeah, and they can also do this thing called antibody-dependent cell-mediated cytotoxicity, or just ADCC, which is way less cool than ACDC. And basically, how that happens is like, the cells have some antibodies. The cells have some like antibody receptors, and if they notice a cell that has antibodies stuck to it because it has like antigens on the outside, that means that the cell is infected. So it kills the cell. Now we are going to briefly mention this little thing that I forgot the name of. Oh, it's a phagolysosome. Okay. So basically, the important thing about phagolysosomes is that they, like, once you phagocyze a bacteria, great, you have a bacteria in you. Now what? You have to kill it. And the way that you do that is through a phagolysosome. It's basically where the vesicle where you phagocyze the bacteria merges with a lysosome, and then you have this respiratory reaction where you use up the oxygen in that area and you use it to make like very harmful gases for the bacteria. And then you kill the bacteria and lyse it up, and then you throw it out like it's poop. And some cells do phagocytosis as like a nutrition thing. I don't think that's the case for immune cells. But just basically think of it as like, remember that scene in Kung Fu Panda 2 where like they're all wearing the dragon costume? This is a really bad dragon. But like, like the guy enters to the mouth and they beat him up and then they like shoot him out. That's that's a phagolyzosome. It's Kung Fu Panda 2. What am I doing? Yeah, that's pretty much it. So now we're gonna talk about adaptive immunity, which is a little harder. So I'm going to go slower. And instead of just explaining it first and then explaining what an MHC is, I'm gonna explain what an MHC is and then explain the rest. So MHCs, just think of MHCs as like a little hand. All cells with a nucleus have this thing called MHC1. MHC1 does endogenous antigen processing, which basically means that if you're a cell that has a nucleus and you have another cell living inside of you, that means that you're a host cell. That's bad. You don't want to be a host cell because once you die, that bacteria is going to spread to other cells, and that's bad. So what you do is you take a piece of antigen and you present it to a CD8 positive cell. CD8 positive cells are killer T cells. So here, MHC1 interact with CD8 positive cells. And basically what you're doing as a cell, if you're doing that, is like you're self-reporting. You're like, kill me before this infection gets worse. Because if you remember when we learned about bacteria that develop inside of other cells, they have lost a lot of their genome that makes them able to survive on their own. So they're there because they can't survive in any other environment. Therefore, if you kill this cell, you kill this bacteria. That's what MHC is one for. That's why MHC is one's for. And it can't interact with helper T cells because you just want a quick and clean death. You're just like, oop, I noticed I got an antigen inside me. Please shoot me. And that's just how it works. However, if you have a cell that is either a dendritic cell, a macrophage, or a B cell, they can do this thing called antigen presenting. They're not all phagocytic cells. B cells are not phagocytic. What B cells do is that they have antibodies on the outside, and when the antibodies bind to an antigen, they just kind of like internalize the antigen itself, slice it up, and present it to a T cell. So cells that have MHC are B cells, macrophages, and dendritic cells. Dendritic cells are the only cells that have both MHC1 and MHC2. All of the other ones have either like one or the other, from my understanding. So, well, not necessarily. Well, just think of dendritic cells as like the bridge between innate and adaptive immunity. I don't like saying that like not any other cell, because when it comes to like biology in general, there are really no absolutes like that. So, yeah. But basically, dendritic cells, when their MHC2 is interacting with a T cell, it's going to interact with a CD4 positive cell, which is a helper T cell. Now, dendritic cells and macrophages and antibodies, if you notice a trend, they're either phagocyzing it or they're getting an antigen from the outside. That's called an exogenous antigen processing, which means that they're getting something from the outside. It's not something that they have internally. It's not like a cell that's living inside them. It's they got this from the environment and they're presenting it to a helper T cell. You would want to present this to a helper T cell because if you have a bacteria inside of you, then all of the cells that notice they have the bacteria, they can just quickly be killed, quick and easy. Just be like, hey, shoot me. And then they get shot and then they get killed. That's great. However, if you find an antigen in the environment, it most likely means that there's a pathogen roaming around, possibly infecting a bunch of cells already. So you'd want to recruit helper T cells because they're the ones that can then recruit B cells and cytotoxic T cells. They can coordinate that whole thing. Cytotoxic T cells can't like just be like, hey, B cells, help me out here. So that's why you would want, if you find something in the environment, you would want to activate a CD4 positive cell, which is a helper T cell. So when you activate a helper T cell, it's gonna, you see this right here? Interleukin. What did I say? Leukocytes talking to one another. Okay, anyways. So basically, it will activate B cells, and it will recruit neutrophils, and it will also recruit cytotoxic T cells, which will then like kill cells with perforins and granzymes, very similarly to how NK cells do it. So like but that's essentially what they do. And then they can differentiate the cells into memory cells. And the usefulness of memory cells is that when you have an infection at first, you have to activate the helper T cell, which then activates the B cell, which then activates the specific cytotoxic T cells, because all of these cells are specific to the pathogen that they encounter. Once you have encountered that pathogen once, when you encounter it again, you don't have to go through this whole bullshit again. All you do is like, this cell that already exists recognizes the antigen and it goes, gotta kill you, and then it like differentiates into plasma cells and stuff. So the reaction is way faster, that's it basically. Oh, another important thing. T cells, you can remember that they're T cells because they mature in the thymus. Well, B cells mature in the bone marrow. So T, thymus, B, bone marrow. Now we're going to talk about B cells. B cells also have immunoglobulin coating their membranes. But the immunoglobulin that they have coating their membranes is usually made of IgD instead of mast cells, which have IgE. And mast cells have a bunch of different types of IgE, while B cells only have one singular type of antibody coating its entirety. So it's like a fuck ton of them, but it's only one type. Well, mast cells have a lot of different types because they want to be broad range because they're innate immunity. B cells are more specific to specific pathogens, so that's why they have only one type. And the important structures of antibodies is that they have this heavy chain right here, and they have this light chain. And then these tips are the variable regions, which means that the only thing that changes within the different types of IgD is like this little tip. And because that's what changes, that's what interacts with the epitope. Just to clarify, let's say you have an antigen that looks like this. If the antibody only binds to this part is called the epitope. So antigen is like the whole thing. Epitope is just the part that binds. I can draw arrows perfectly. The epitope is the part that binds actually to the immunoglobulin. So you also have 5 types of immunoglobulin. You have IgG. You have IgD, which I mentioned, which is like the thing that coats the B cells. You have IgG, which is the one that works with C3b, which does opsonization, and it also does this thing called neutralization, but I'm going to go over that later. You also have IgM, which is like when you have a response, you usually make IgM first, and then you switch into making IgG. Not sure exactly why. That's just how it is. You have IgA, which are like, they're the only secretory immunoglobulin. So the way that I remember it is like, if you have a baby, if the baby's inside you, the baby is getting immunity through IgG. However, once the baby is out of you, it still needs immunity because it's a baby. It has no immune system. So the way that it gets immunity is through IgA, through breast milk. So it's secretory, so it's like in the mucosal surfaces. And if you don't know, mucosal surfaces are just like anywhere in the body where you have a hole and it's moist. That sounds weird as fuck, but that's just how it is. That's a mucosal surface. So all of those surfaces like secrete IgA because you want those surfaces to be the cleaner ones. Because obviously if you have like a big gaping hole, you want it to be clean and free of pathogens, because if pathogens keep getting in, that's like bad for you. You don't want that. So, and you also have, as I mentioned, IgE. And that one is to deal with either like allergic reactions or they're the ones in mast cells. So this chart right here talks about the difference between primary response and secondary response. Secondary response is memory cells. So the memory cells, they don't have to be differentiated again. They don't have to be like coordinated by the helper T cells. So they already know what to do. So they just like make a fuck ton of antibodies, make a lot of IgG already from the get-go, and then they kill off the pathogen. This is way faster than the first time around. The first time around, first you make IgM and then you make IgG, and it's a pain, and you don't want that. And also, the titer, which is like the amount of antibodies that you produce, is lower for the primary response because of the fact that like you're getting used to like dealing with a pathogen, and because this deals for much longer, you have to make like much less immunoglobulin because you do it for a longer amount of time. But the secondary response, it's much faster. It's like there's a higher titer right from the get-go. So that's just how it goes. So when you activate a B cell, you can either do it T cell dependent activation or T cell, oop, I forgot a picture. You can either do T cell dependent activation or T cell independent activation. Let me see if I have a picture for that. I don't? What the hell? Well, basically, T cell dependent, let's talk about T cell independent first because it's easier. T cell independent, it's just like you have an antigen that is T cell independent that triggers the B cells, and then it just does it on its own. So it's faster, but it's less effective. And then when you have T cell dependent B cell activation, then you have to have the signal from the antigen, which is like the epitope binding to the antibody. And you need the signal from the cytokines coming from the helper T cells. So if you have those two signals, then boom, you're done. It's like how T cells have that two-factor authentication thing, which I also didn't put a picture of that. Weird. But basically, T cells have this thing where they have like the MHC interacting with the T cell receptor. They also have like a little signal, and they also have cytokines. The importance of this over here is that when you're making T cells, they go through a lot of selection. And then like they go through this thing called positive selection and negative selection. Positive selection is just making sure that it can identify MHCs. And negative selection is if it identifies like an MHC that doesn't have any antigen on it and it still like goes against it, then that cell is killed. But again, with every regulation practice, there's always something that can be a mistake. There's always like a way that you can let a cell slip. So if you do have a cell that interacts with an MHC but doesn't have this second signal, then you just turn that cell off. And it just stays there, just like hanging out. Which is useful for you because remember how I said the MHCs are like hands? Imagine that the T cells like shake hands with all the MHCs. So if you're a cell with MHC type 1 and you notice that you have a pathogen in you, then you would like present the pathogen in your MHC, and then the T cell will shake hands with you, and the T cell is going to be like, oh, you're infected. Pew! However, if you have a cell that doesn't have anything on its MHC, and the T cell interacts with it and is still like, oh, I'm going to kill you, that's bad, because obviously you need cells in your body. So if the T cells just kill all the cells, that's not okay. So that's what these secondary signals are for, because you want to make sure that the cells that you're killing, you actually have to kill. That's a similar idea as to why you have T cell dependent immune responses with B cells, just so you make sure that like you're actually producing antibodies when you need to, so you don't waste energy. So on the topic of antibodies, they work, why is this picture so small? They work in very specific ways. So they can do this thing called precipitation, which is like when they bind a bunch of antigens and it makes them settle out of the solution so that they can't like hurt anyone anymore. They can do agglutination, which is like when they stick to bacteria and make them all stick together so that when like a neutrophil goes to phagocyze it, it can eat a fuck ton of them at the same time, which makes the reaction goes by so much faster. Because if you think about it, bacteria, they reproduce really, really rapidly. So you'd want to like end them as soon as you can. They can do opsonization, which I mentioned, that it's like IgG and C3b. And also it in like, this would be IgG, for example. It just makes it easier to identify for you to phagocyze it. This right here, this doesn't mean that the antibody itself lyses the bacteria. This is just like, remember how the classical pathway, it's dependent on antibodies? That's what it's happening. This is just a complement system. And they can also do this thing called neutralization, which is like when it blocks either a toxin or a virus, it blocks their adherence factors. So if you remember how a virus works, it like lands, adheres, and then it injects. If it can't adhere, then this shit never gonna happen. That's great for you. You don't want viruses infecting your cells. So that's how neutralization works. This is a pretty comprehensive overview of what you need to know. I know that there are a lot of slides that have a lot of detail, but I wouldn't recommend studying all of the detail. If you understand the main reactions and the main like pathways of immunity, then I would say you're relatively fine. Because for the exams, I never memorize all the slides. I just know, if you know the reactions, then everything else just falls into place. So like when you're, let me scroll. So like when you're memorizing all the cells, if you know what the path is, then all of the cells eventually fall into place. Except for like eosinophils and basophils. You don't really see them in like the average. You know what I mean? Like they're kind of weird. But like all of the other cells, once you know the flow of information and the flow of the reactions, then all of the cells eventually fall into place. And you don't have to like spend time being like, oh, macrophages do this, mast cells do this. You just kind of like are able to remember from the reactions. So what I would do if I was struggling with immunity is I would try to draw out the initial acute inflammation that happens. I would draw it, and then I wouldn't label anything. I would label what's going on, and then I would label the cells, because then it will make it easier for you to remember what each cell does. But I'm sorry if this was kind of rushed. I'm tired. But if you guys have any questions, just let me know, and I got you. Like, you can message me privately, and I hope this helped.